Welcome to Longevity Papers Podcast. Each week, we explore the latest scientific papers, pulling out the key insights for you, our listeners, in biotech research.
And look, let's just be upfront. This week, honestly, like most weeks in aging research, it really hammers home that maybe 95% of the papers coming out are, well, incremental steps.
Right.
They're not the massive paradigm-shifting breakthroughs everyone's chasing. You see a lot of mechanistic work in worms or mice, biomarker studies, small interventions.
Important work, but maybe not life extension breakthroughs.
Exactly. They're usually not the kind of thing that's going to suddenly add 10-plus years to human lifespan. That's still the moonshot.
It's the reality, isn't it?
Yeah.
But even with that landscape, we did find a few papers this week that, made us pause. We're definitely looking at them with skepticism, as always.
Absolutely.
But they offer some interesting angles. Maybe touch on known pathways in a new way,
Mm.
or, in one case, introduce something totally novel. So today we're going to unpack these. We'll try to tease out what's genuinely new versus what's just another small step, and really help you understand the potential, but also, crucially, the limitations right now. Let's get into it.
Okay.
First one. This paper's titled Chronic Activation of a Key Exercise Signal Transducer, CANMKII, Drives Skeletal Muscle Aging and Sarcopenia.
Yeah.
The authors are M.R. Binet, T. Chung, and W.A. Fountain from Johns Hopkins School of Medicine. It was posted on bioXFuv, that's pronounced bioRxiv, August 2nd, 2025.
Got it.
And it's super relevant because it's tackling muscle aging, sarcopenia, and maybe hints at ways to intervene.
Yeah, this one caught my eye because of the core idea, this antagonistic pleiotropy thing in muscle. Sarcopenia, that age-related muscle loss and weakness, it's a huge problem for healthy aging, right?
Huge.
We all know exercise is crucial, and part of how it works is by briefly switching on these signaling pathways, calcium and ROS-dependent signals. CAMPI is a big one, calcium calmodulin-dependent kinase the second.
Right, the exercise signal.
Exactly. Short bursts are good. They boost muscle performance, help it adapt. But here's the kicker the paper explores. Those same signals, especially CAM-KI, seem to get stuck in the on position in older muscle, chronically elevated.
So too much of a good thing.
Precisely. Instead of helping, this chronic activation actually drives decline. It's like the perfect example of antagonistic pleiotropy, something beneficial when you're young, like these adaptation signals, becomes actively harmful when it's just constantly running in the background as you age.
And the results seem to really back that up. They made young mice with this CAMKII constantly active, like in old muscles.
Yeah.
And these young mice started looking old, muscle-wise. Their muscles couldn't contract properly. They started wasting away. Mitochondria were disorganized, powerhouses.
Wow, okay.
They even saw these things called tubular aggregates, which are apparently classic signs of damage in aged muscle fibers. And it wasn't just physical. Their gene expression profile shifted, became more inflammatory, more stressed.
So it really mimicked aging just by flipping that one switch chronically.
Seems like it. And they even think they found a mechanism for the weakness. Something about altered heme metabolism, separate from the muscle shrinking.
That's pretty neat. And presumably they tried inhibiting it in old mice.
They did. They found that partially inhibiting CAMKI in aged muscle actually improved how well the muscles contracted.
Partially. Not even full inhibition.
Right, just partial. And it nudged the gene expression back towards a younger profile. Interestingly, it did this without making the muscles bigger. No hypertrophy.
Okay, that is interesting. It's definitely one of the more compelling mechanistic papers this week, mostly because it shows that antagonistic pleiotropy so clearly. But let's temper the excitement a bit for our listeners in biotech. This whole pattern, good acutely, bad chronically, it's a classic theme in aging research, isn't it?
We see it a lot.
We do. And while these CAMKI inhibition results are, promising for muscle function, they're limited. There's no lifespan data here.
Good point.
Does fixing the muscle function actually make the mice live longer? We don't know. Does this effect extend beyond muscle? Maybe impact systemic aging? Also unknown.
But how would you even dose it, pulse it, low dose constantly?
Exactly. Tons of questions remain. For biotech, maybe this points towards needing really smart drug delivery or modulators that can tune these pathways, not just switch them off. Context is everything.
So maybe a piece of the puzzle, but not the whole picture.
I think that's right. It feels like it could be part of a bigger strategy down the line, but it's not the magic bullet on its own.
Okay, let's shift gears from muscle to something more systemic cellular senescence. Paper number 2. Targeting cyclin D1-CDK6 to mitigate senescence-driven inflammation and age-associated functional decline.
Senescence, always a hot topic.
This one's also from bioXFuv, August 2nd, 2025. Authors are A. Rajesh, A.P. Havas, and R. Arnold at Sanford Burnham Previs. And the relevance is obvious: senescence, aging, inflammation, potential therapies.
Right. So the background here, which is probably familiar to many listeners, is that senescent cells, these zombie cells, drive a lot of aging problems through chronic inflammation. They pump out this inflammatory soup called the SASP.
Senescence-associated secretory phenotype.
That's the one. And often involves these interferon-stimulated genes, or ISGs. It creates this nasty pro-aging environment. Now, the weird thing, the paradox, has always been cyclin D1, CCND1. It's usually involved in making cells divide.
But senescent cells don't divide.
Exactly. Yet CCND1 levels are high in them. So what this paper proposes and shows pretty convincingly is that CCND1 and its partner kinase CDK6 are actually driving the SASP and ISG expression.
How?
By basically messing with the DNA. They seem to promote DNA damage accumulation. This damage leads to bits of chromatin breaking off and ending up in the cytoplasm, these cytoplasmic chromatin fragments, or CCFs. And those CCFs then trigger this pathway called CGO sting. Normally, CGA sting is part of the immune response to viruses detecting foreign DNA, but here it gets activated by our own DNA being in the wrong place, and that kicks off a massive inflammatory response.
Wow. So it hijacks an immune pathway.
Sort of, yeah. And they also found that P53 and P21, the tumor suppressors, normally try to put the brakes on this CCND1 CDK6 activity.
And what did they find experimentally? Did it hold up?
Seems so. They looked in aged mouse livers, found higher CCN1 in the senescent liver cells, the hepatocytes.
Makes sense.
Then they either knocked out CCND1 just in those liver cells, or they treated the mice with a drug called palbociclib.
Palbociclib. I know that name. Isn't that a cancer drug?
It is. It's a CDK46 inhibitor. And yeah, in these aged mice, blocking CCND1 or using palbociclib reduced the DNA damage and those inflammatory ISGs in the liver.
Okay, that's mechanistically neat. But did it do anything for the mice overall?
That's the exciting part. Treatment with palbociclib actually reduced frailty and significantly improved the physical performance of the aged mice.
Okay, that is interesting. Improving frailty is a big deal.
Huge potential, right? Because, like you said, palbociclib is already an FDA-approved drug. That gives it a head start. The mechanism they outline connecting CCND1 CDK6 to DNA damage, CCFs, CGS stinging, it's a really strong story. And seeing functional improvements like reduced frailty is always encouraging.
I sense a but coming.
Oh, there's a big but. Several, actually. First, palbociclib is a cancer drug for a reason. It has significant side effects, serious toxicity, like hitting bone marrow function. Using it chronically for aging, that's a massive safety hurdle, huge.
Right, you can't just give everyone a cancer drug indefinitely.
Absolutely not, without extreme caution and validation. Second, again, no lifespan data in this paper. Do these improvements last? Do the mice live longer and healthier, or is it just a temporary boost? We don't know.
And is it fixing the root cause or just masking the symptoms?
That's the million-dollar question, isn't it? Is it preventing senescence, clearing senescent cells, or just dampening their inflammatory output? For biotech, this is still really interesting because it points to a clear, druggable target, CDK46, involved in the bad side of senescence.
Maybe more targeted delivery. Or different dosing.
Perhaps. Pulse dosing, intermittent therapy, combination approaches to lower toxicity. There are avenues to explore, but the critical next steps are obvious. Proper lifespan studies, deep dives into chronic low-dose safety, and figuring out if it's truly senolytic or just xenomorphic, changing the senescent cell's behavior.
Yeah.
Fascinating pathway, but a long road to translation for aging.
Okay, we've hit mechanisms, druggable pathways. Let's go somewhere completely different for our last paper. This one's called Cyrene, a novel geroprotective compound that extends lifespan and health span in C. elegans and Drosophila.
Cyrene. Never heard of it in this context.
Me neither. It's from A. Alokda, S. Yadav, and A. Casas at McGill University, posted on bioRxiv, August 1st, 2025. It's intriguing because it's a totally new compound, claiming longevity benefits in 2 different model organisms.
Okay, novel is definitely the word. Cyrene. Dihydrolevoglucosanone. I mean, this stuff is mostly known as an eco-friendly solvent in chemistry.
Right. Not your typical longevity drug candidate.
Not at all. So seeing claims of cross-species effects C. elegans and Drosophila plus broad stress resistance, yeah, that makes you look twice. But I have to say, my skepticism alarm is going off pretty loudly already on this one, like really loudly.
Okay, let's see why. The paper says Cyrene extends lifespan and health span in the worms, C. elegans, Better movement, resistance to all sorts of stress, oxidative heat, osmotic DNA damage, protein clumping.
Sounds almost too good to be true for a solvent.
They even saw some protection in worm models of neurodegeneration. But here's the catch, and it seems like a big one. It only worked if they gave it to the worms during development or really early adulthood. They specifically say it had to be given before day 8 to extend lifespan.
Ah, before day 8. That's telling.
And mechanistically. They say it doesn't depend on the bacteria the worms eat, and it's only partially independent of DEF16, that big FOCOSO transcription factor pathway for longevity.
Partially independent.
It did also extend lifespan and boost stress resistance in fruit flies, Drosophila, so there is that cross-species conformation, but that early treatment window in worms.
That early window is exactly why my skepticism is through the roof for any kind of mammalian translation. Day 8 in C. elegans is still very young, practically developmental.
So you think it's affecting development, not aging?
It screams developmental robustness to me. It suggests Cyrene might be making the young worms tougher, more resilient from the start, rather than actually slowing down the aging process later on. That's a huge difference if you're talking about intervening in aging humans.
Yeah, giving something to a developing worm is not the same as treating an older adult.
Not even close. And the mechanism being fuzzy, only partially independent of DEF16, that doesn't help. It means it's not clearly hitting the major longevity pathways we understand best. How do you even begin to translate that?
Good question.
And look, let's be brutally honest. The number of compounds that look great in worms and flies and then completely fail in mammals is enormous. It's the graveyard of longevity drug development.
Sure.
Without a clear mechanism, and with this a massive question mark about whether it's even targeting aging versus just development or general stress resistance, frankly, this could just be statistical noise. Maybe it coincidentally worked in these two specific models under these specific conditions.
So what would need to happen next for this to be taken seriously?
Oh, a lot. Definitive mammalian studies, obviously. A much clearer idea of the mechanism, especially the DAF16 independent part. And solid proof it's actually modulating aging processes, not just making organisms generally tougher early on. Until we see any of that, this is, well, it's a curiosity, an oddity from the solvent shelf.
Okay, so summing up the week then.
Yeah, I mean, tying it all together, this week feels like a perfect illustration of that 95% statistic we started with.
Right.
Most of what we see, even the interesting stuff like the CAM-KII or CDK6 papers, while valuable for understanding mechanisms, they aren't delivering those huge leaps in lifespan extension that the field is ultimately aiming for. They're refinements, important ones maybe, but still refinements.
Yeah, we're seeing these really detailed mechanistic studies identifying pathways, showing some modest functional benefits or hitting known hallmarks like senescence, but nothing feels truly revolutionary in terms of extending maximum lifespan by a significant chunk.
Exactly. It feels like we're still very much stuck in the mode of identifying and tweaking individual aging hallmarks, chipping away at the edges, which is necessary work. But it's not the same as finding a fundamental reprogramming approach that would really shift the whole paradigm.
Fine-tuning the engine versus inventing a whole new way to travel.
That's a good way to put it. And it leads to a really important question for everyone listening, for the whole field, really. We're good at optimizing existing pathways, managing hallmarks, but what kind of discovery, what kind of breakthrough would actually force a model change in how we even think about aging? What would that look like beyond just getting better at fiddling with the known knobs?
Something beyond the current framework entirely.
Yeah. What's the fundamental insight we're missing that would allow for true biological reprogramming, not just incremental gains?
That is definitely something to chew on. A critical question for all of you working in biotech. Thanks for joining us as we unpack these latest papers. We'll be back next week with more research, more insights, and, you can be sure, more critical analysis.